All right, y'all, let's go ahead and get started. How's everybody doing? Everybody happy to be in school on President's Day? That's why they have the day off. Yeah. Okay, so we have basically finished up our intro to all of the different classes of biomolecules, right? So we talked about how proteins form. We talked about the formation of polypeptide chains, making peptide bonds. We talked about the properties of amino acids. We then moved into nucleic acids, carbohydrates, lipids, and now we're coming back to proteins, okay? But we're now focusing on proteins from the perspective of thinking about how they serve as catalysts for reactions, okay? So proteins are not the only biomolecule that can do this, right? RNA can easily catalyze reactions. That's the ribosome and lots of other things. As it turns out, carbohydrates actually can too in some cases. But enzymes are by far the biggest workhorse of the cell, right? Basically, the primary function of proteins is to serve some specific purpose, and usually that is to catalyze a reaction. Okay, so today what we're going to think about is what we mean by catalyze a reaction, and then go through some examples. Okay, things to really remember from our intro to proteins is that when the polypeptide chain forms, right? So like as we make this long polymer of amino acids, it adopts secondary, tertiary, and quaternary structure, which ultimately creates a three-dimensional scaffold. And that scaffold is what then creates an opportunity for these proteins to recognize very specific things, bind to specific things, and then catalyze reactions. Okay, so if you just remember that the protein itself provides this structural scaffold to bind things and to facilitate a reaction, that's the most important thing moving forward. Also, remember the amino acid side chains, and there's some of them that are really important with specific functional properties. Things like the ones that can serve as general acids or general bases, right? So our two acidic amino acids are what? Huh? Okay, so glutamic acid, yep, glutamate, and aspartic acid, right? Those are the two acidic ones. What are the two primary basic ones? So histidine is technically grouped as a basic one, but it's not a very good basic amino acid. What are the other two? Lysine and arginine, right? So lysine and arginine are the two big basic ones because they are always protonated under physiological conditions, okay? So the idea being that if you need an acid or a base, you should be thinking about those amino acids, not to say they're the only ones that can't do it. And I specifically point this out because we're going to see today that one huge feature of enzyme catalyzed reactions is the utilization of general acids and general bases. Generally speaking, if you need an acid, if you need to give a proton to a reaction, it is there. Propose there's a general acid, propose there's a lysine or an aspartic acid or a histidine or something acidic that can give a proton. If you need a base, it is there. Choose an aspartic acid, choose a lysine, choose a histidine, something that can abstract a proton from the molecule you're working with, okay? So with that said, let's go ahead and dive in. All right, so enzymology is a massive field in biochemistry. It is this massive field that studies enzymes, right? And there's really kind of four big things that enzymes do. The first one is that they increase reaction rates, okay? Meaning they're catalysts, right? The point of a catalyst is to increase the rate of a reaction. And does anybody know how, anybody remember how catalysts work? How do they increase the rate of a reaction? Yeah, they decrease active. Oh, listen, in Gen Chem, all right. They decrease activation energies, right? That's how enzymes work, right? So if we think back to kind of basic principles of chemical reactions, thinking about on the x-axis here as the progress of a reaction, proceeding from reactants to products. On the y-axis is just going to be, generally speaking, energy. Okay, we can think about a relative energy, Gibbs free energy, of our reactants. We can think about a relative Gibbs free energy of our products. Right? And the fact that I made the products lower in energy than the reactants suggests that this reaction is spontaneous, right? When the products are at lower energy than the reactants, this means the reaction is spontaneous. That tells you about the thermodynamics. It tells you nothing about kinetics. The kinetics boil down to how much energy it takes to get over the energy gap between the reactants and this transition state, right? So this energy right here is our activation energy, which we describe as EA. Hopefully you remember this to some point, right? And this point right here is this, it's called the transition state. It is the highest energy point that proceeds during a reaction, okay? And if we think back to kind of the general carbonyl chemistry that we talked about at the very beginning of class on day number 1, right? So you have some nucleophile. We'll just say it's a deprotonated alcohol group. You have a carbonyl. Okay, so in this mechanism, a lone pair on this deprotonated nucleophile attacks the electrophilic carbon. We generate this intermediate. And the reason that we always draw this intermediate is because this intermediate is the highest energy point on a reaction, right? So you have this intermediate where we have an oxyanion on a tetrahedrocarbon. This is the transition state. This is the highest energy point on that reaction diagram, okay? So the way that enzymes work is that they decrease this activation barrier, okay? So, and we're gonna see in many, many cases, the way that generally they do it is by positioning some positive charge at the point where this oxyanion forms, okay? So by positioning this oxyanion close to some positive density, then that creates a nice electrostatic or ion-ion interaction between the transition state and this positive patch in the structure of the enzyme, right? This favorable interaction will take this massive activation barrier that we showed here and decrease it. And by decreasing the activation energy, we increase the rate of the reaction. I'll show this equation up here. If you don't remember, that's fine. You don't have to remember moving forward. But remember, the rate constant is equal to this property A times e to the minus EA over RT. And the idea here is K is the rate constant. The smaller K is, the slower the reaction is. And it's related to this activation energy, right? So we raise e to the power of negative EA, which means the smaller the activation energy is, the faster the reaction is. And hopefully that makes sense, right? So the idea is that we need to give our reactants enough energy to get over this bump, right? And in Gen Chem, I describe it as like climbing up the ladder to go down the slide, right? You have to climb up the ladder to get to go down the slide. You have to invest energy for the reaction to happen. You can get to go down the slide a lot faster if it's a smaller ladder to climb, okay? So this is the way enzymes work. As you're thinking about enzymes, this is the kind of things you should think about. And the reason this is so relevant and so important is think about everything that has to happen in a cell. In E. coli, right? There are literally thousands of reactions that have to happen for the cell to be able to divide. Thousands of reactions. You have 3 million base pairs in E. coli's genome that have to be replicated for a cell to be able to divide. Yet E. coli can do that in less than 20 minutes, right? That's pretty remarkable. That wouldn't happen if you just threw everything together, right? It's because enzymes are so good at decreasing the activation energy, at increasing the rate of DNA polymerization, that this can happen in the time frame that it needs to. Okay? It decreases the requirement for really harsh conditions. In orgo, you talk about doing things in organic solvents and really making the conditions quite harsh, right? Making, like adding a bunch of hydrochloric acid or adding a bunch of sodium hydroxide, making the conditions really, really harsh that would absolutely not promote formation of these reactions in vivo, right? We cannot create the harsh conditions that would otherwise be required for these enzymes, these reactions to happen. A couple examples here. Think about what we need to happen to form a peptide bond, right? Remember, the peptide bond forms when you have the amino group of one amino acid condensing with the carboxylic acid group of another, right? And for this amino group to be a good nucleophile, it has to be deprotonated. So we need to have an NH2, not an NH3+, because we need to have this lone pair being reactive. Remember the pKa of these amine groups? The pKa of the side chain of lysine, the pKa of the N-terminus of amino acids is all up in the 9 to 10 range. But physiological conditions are down in the 6 to 7.5 range, right? So this amino group on the N-terminus of the amino acid is always protonated. So how can you deprotonate this under physiological conditions so that it can serve as a nucleophile to allow the formation of a peptide bond? That just wouldn't happen, right? Unless you can create a chemical environment where this can be deprotonated, which means, quite frankly, excluding it from water, creating a new chemical environment so that this can be deprotonated. And that's one big important role that enzymes serve, is that they create these microenvironments that specific chemistry can happen in. Okay, another one is methylation of uracil. We won't really talk about that, but really, so think about taking a methyl group, just taking methane and trying to throw it onto the base group of uracil. It's not going to be something that's going to be favorable under any conditions unless you can create this microenvironment that lets it happen. Okay, another really important one is conferring specificity, right? So think about the hundreds and perhaps thousands of compounds floating around a cell at any given time that look really similar. Think about our carbohydrates, right? There was glucose, mannose, and galactose, all of which differed only by the orientation of a single hydroxyl group on one of the carbons, right? Going from glucose to galactose, all you did was flip the orientation of the hydroxyl group on carbon 4. That's the only difference. So if you need an enzyme to be able to function only on galactose and not glucose, or only glucose and not galactose, they need to be able to recognize that really small, subtle difference, right? And that's one thing enzymes can do really, really well. Another example here is a salivary enzyme we have, like as we start digesting sugars, is called amylase, alpha amylase, right? It's present in our spit, basically. When we start chewing on sugar-containing molecules, this enzyme goes to work and starts breaking down sugars. It breaks down starch so that we don't have to break down as much further down the digestive tract. But it does not work on cellulose, right? Try chewing bark or try chewing grass, right? It doesn't work because cellulose is made of a beta linkage between glucose units and not alpha linkages. This enzyme only recognizes the alpha linkage between glucose units on sugar polymers. And this is the last big one that we probably won't really get into in this class, but it enables control over the chemistry. Okay, and this is something called allosteric control, right? So the idea being that we want to be able to turn the activity of the enzyme on or off depending upon specific chemical environments or specific conditions. And one really good example of this, right? Think about metabolism. Okay, think about glycolysis. Everybody at least generally know what glycolysis is. If you don't, you will in a few weeks when we talk about it in class. But so you have this pathway where we start with glucose and we break it down to pyruvate to make energy. Well, we don't want these enzymes to necessarily be to be functional if the cell already has massive amounts of energy, right? If we already have an abundance of ATP in the cell, we already have way too much energy. What's the point of taking that sugar, breaking it down to give us more energy when we already have too much? So the idea is that there's a couple key checkpoints throughout glycolysis where it can recognize high ATP concentrations. And when those concentrations are high, it tells the enzyme to stop working through something called allostery. So it's the idea that an interaction at one part of the enzyme propagates that signal through the rest of the enzyme and makes the active site less active. All right. So with all that in mind, I already mentioned this, right? So we kind of talked about this. The idea of how the enzyme increases reaction rates is it decreases the activation barrier, right? That's the way catalysis works. Every catalyst, no matter what it is, the way that it works is it decreases the activation energy. and that then creates a faster reaction. Okay, specificity. Here's one example that we're actually going to see a little bit later as well. So proteases, right? Digestive enzymes again. We have a bunch of these digestive enzymes that are present in our in our digestive tract. They're secreted by the pancreas, and they recognize specific amino acids within a polypeptide chain. So some of them will recognize a long basic group, right? So things like arginine or lysine. Some will recognize big hydrophobic groups. Chymotrypsin does that. And some of them recognize small hydrophobic groups. The idea is that right by the active site, there's this specificity pocket that looks for something specific. Trypsin looks for a long chain that terminates with a positive charge. Chymotrypsin looks for something that's big and hydrophobic and can fill this big space. Elastase looks for something small and hydrophobic. If something big and hydrophobic just won't fit there, but something small and hydrophobic will. Right, so the idea is that a scaffold that's produced by the enzyme is looking for specific things or excluding specific things, right? So the example of elastase, this recognition pocket is too small to fit something big and hydrophobic. So it will only recognize small things like alanine, valine, or glycine. Trypsin, at the bottom of this channel, at the bottom of this groove, it would have a concentrated negative charge. So it's specifically looking for the ion-ion interaction that will happen between the positive charge on lysine or arginine and that negative charge. If something binds to the enzyme that can't make that interaction, it won't be recognized. It won't bind tightly enough to react. Okay, and there are lots of these types of examples that we can go through. We clearly can't go through all of them, but as you're kind of thinking about how enzymes work and as you're learning about it, just think about specific functionality that one substrate has over another and think through what the enzyme might present and or not present that could recognize one thing over another. Okay, here's another example is this idea of stereospecificity. I mentioned something being able to recognize galactose and not glucose, or glucose and not galactose, right? And the only difference is one stereocenter. So the enzyme must have this ability to specifically recognize the position of one functional group and not another, right? So if you think about just this one chiral carbon here, here we have a hydroxyl group positioned at this position and a methyl group positioned here. Okay, so if an enzyme were going to recognize this stereocenter, you could certainly imagine it positioning a hydrogen bond acceptor or donor right here and something that's hydrophobic right here so that it specifically recognizes this stereocenter. And if it were switched, if the methyl group were here and the hydroxyl group were here, they wouldn't bind tightly. They would actually exclude each other. They would prevent each other from binding because something that's polar wouldn't want to interact with something that's hydrophobic. Something that's hydrophobic wouldn't want to interact with a hydrogen bond donor or acceptor. So enzymes can recognize stereocenters based on specific intermolecular forces positioned at very specific places. Questions so far? Okay. All right, so now we're going to move into our classes of enzymes, and we're going to go through 2 more slides, and then I'm going to give you about 5 or 10 minutes to work on the first page of your activity. Okay, so there are 6 classes of enzymes, and what I think is most remarkable about this is the fact that there are hundreds of thousands of chemical reactions that happen in any given cell. There's a massive number of reactions, but we can break all of these down into 6 classes. Okay, and being able to recognize patterns like that makes something that's really daunting into something that's a lot simpler, right? And that's really kind of the goal of this, is to make something that's very daunting, characterizing hundreds of thousands of reactions, and then breaking it down into this just really need to understand 6. Okay, the first one is oxidoreductases. As the name suggests, these enzymes catalyze oxidation and reduction reactions. And remember, oxidation and reduction reactions always happen together. So if something gets oxidized, something else gets reduced. And it's really, really, really common with these that you're looking for specific cofactors, specific molecules that partner with the enzyme that are able to donate or accept electrons. Okay? From other classes, does anybody know one or more of these common cofactors? Think about glycolysis, the TCA cycle, and oxidative phosphorylation. What's the molecule that's produced during glycolysis and the TCA cycle that then goes on to carry electrons to the mitochondria? NADH and FADH2, right? So these are two really, really big redox active cofactors that really without these, we can't catalyze electrochemical reactions. So if you're looking at a reaction and you see NADH or FADH2 part of the reaction, it is oxidoreductase because these are redox active cofactors. Transferases are class 2. The idea here is you pick one functional group off of one molecule and move that entire functional group to something else. Okay? Anything that moves like a phosphate from ATP onto a different substrate, that would be a transferase. Anything that moves a methyl group from one substrate onto something else would be a transferase, right? So it's not just breaking a bond, it is taking one full functional group and moving it to another molecule. Not to be mistaken for moving it to somewhere else within the same molecule. You have to transfer it from one molecule to another. That's a transferase. Hydrolases are one of two classes of enzymes that break covalent bonds. Hydrolases break covalent bonds by using water. So they add water across the bond. So if you see one thing becoming two and water is a reactant, that's a hydrolase. Okay? So over here, right, so like we're talking about this condensation reaction between an alcohol and a carboxylic acid. So the reverse of that would be using water to break that bond. That's hydrolysis. That'd be catalyzed by hydrolases. Lyases are related. They break covalent bonds, but they do not use water. Okay, and one theme with lyases is that you'll always create double bonds or triple bonds in theory. But if the bond you're breaking is single, an adjacent bond will end up being a double bond, right? Because the electrons have to go somewhere. So if you break a bond and you have not used water, that's a lyase. The reverse of not quite yet. An isomerase is kind of like a transferase. You are moving one functional group somewhere else, but it's within the same molecule. So you're creating an isomer. This could be converting glucose to galactose, right? You're rotating the bond on carbon 4. That's making a stereoisomer. That would be an isomerase. You could be taking a methyl group from carbon 6 of something and moving it to carbon 3. That would be an isomerase. You are creating an isomer. It can be stereoisomers or structural isomers. But the idea is you have one substrate and you change how things are positioned in that substrate. And the last one is the reverse of hydrolases and lyases. Hydrolases and lyases break bonds, ligases join them together. Right? So DNA ligase, it's in the name. It takes two pieces of DNA and fuses them together. So if you create a bond, you go from two things to one thing, that's a ligase. All right, so then the last slide here before I give you some chance to work on the activity is I mentioned that there are some important cofactors, and this is not by any means an exhaustive list. These cofactors here, these are important for, so I guess the definition of a cofactor is a molecule that's really important for an enzyme catalyzed reaction, but it's not actually part of the polypeptide chain, okay? And we could break it out into more specific terms, but if you understand just the general idea of a cofactor, then you're good to go. They come in two forms, metal ions or organic molecules. Okay, and the organic molecules are often called coenzymes. I don't care if you know the differentiation, honestly. The idea is these are molecules that are critical for the function of an enzyme, but are not part of the polypeptide backbone. Okay? Some common metal ions. Copper and iron are usually really important for redox reactions because they're able to cycle between a +2 and a +1, or a +2 and a +3 charge state, right? These are the two metals that are available to switch between charge states within the restrictions of a cell. Magnesium, manganese, nickel, and zinc are not. They all have fixed charges, right? Go back to Gen Chem where we talked about like the variable charge metals and the fixed charge metals. Copper and iron are variable charge. The rest of these are fixed charge, at least in the context of a cell. Okay, so they're going to be more structural in nature. They're going to make sure the enzyme adopts the proper structure. They're going to help to make sure that the backbone of ATP, that negative backbone, is positioned in the correct way. The organic cofactors, the coenzymes, these are the ones that typically are really important in the chemistry. Okay, so we have NAD, NADP, FAD, FMN, CoA, ascorbic acid, etc. All right, so I'm going to give you let's say till 8:35 to go ahead and work through the first page, and you might be able to get done a lot faster. This is just like looking at a reaction, see if you can recognize what the reaction is, what class of enzymes, and then think about these cofactors and think about which one of them are important in oxidative reductases.